Hello, everyone, and welcome to today's live broadcast, How Biomarker Testing Shapes Immunotherapy, presented by Dr. Condie Carmack, Vice President of Precision Oncology, Vela Diagnostics. We are excited to bring you this educational webcast presented by LabRoots and sponsored by Vela Diagnostics. For more information, visit www.veladx.com. I'm Julie Simmeroth of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button on the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. Please join me in welcoming Dr. Condie Carmack. I will now turn the presentation over to him.
Well, great. Thank you, Julie. After a long research and development phase, anti-cancer immunotherapies have burst onto the oncology scene and are producing some remarkable results. And they promise to get even better. Less than 10 years ago, the field consisted of true believers, immunologists who were convinced that the immune system could be taught, tricked, cajoled, bribed, lured, seduced into mounting an immune response against tumors. There were a handful of anecdotal cases that kept the believers going, just enough to keep the funding and the passion alive. Things have dramatically changed with rapid developments in terms of scientific understanding and clinical outcome. The identification of cancer-specific antigens recognized by immune cells and the elucidation of inhibitory pathways that lead to crosstalk between neoplastic and immune cells has moved immunotherapy from phenomenology into predictable and dependable standard of care medicine. This is not just another cancer drug. This is an entire industry of treatment. It really goes to the heart of personalized medicine and is now representing personalized therapy. I will talk about both the good points and the bad points of the technology. This is going to change the way that we treat oncology forever. So here's my agenda for today. I'm going to cover the history and the background of the immunotherapies. They've been a long time in coming, and not all of them are success stories. We're going to review the categories of immunotherapies. I'll talk about the mechanism of action, or what we think is the mechanism of action. I'll also cover the major biomarkers of the ITs. I'll also talk about the progress in clinical testing, and I'll touch upon the adverse reactions of some of the ITs, along with some of the success stories. As way of background, as I indicated in my introduction, research in the immunotherapies is nothing new. People have believed for some time that they could stimulate the immune system with nonspecific agents, so-called immunostimulants. These, for the most part, have consisted of bacterial toxins and cell components. It has met with limited success, but this actually led to the next generation of recombinant cytokines, like interleukin-2 and alpha-ferron. Some of these cytokines are still used today. Monoclonals have always been a staple of immunotherapy, but identifying cancer targets that were unique to cancer cells has been an issue. Monoclonal antibodies have been used in cancer treatment for over 25 years. The poster child, of course, is trastuzumab or Herceptin, which dates back to the early 1990s. Prophylactic vaccines have also been around for decades, but now with the combination of immunotherapies, they're poised for a resurgence. And last but not least are the immune checkpoint inhibitors that have been shown the most promise to date. These include ipilimumab, or IPI for short, pembrolizumab, or Pembro, nivolumab, or Nivo. These are three of many of the immunotherapies. There are lots of others, but I'm going to cover the big three right now. And I'll cover these as we go along. So in this slide, I just wanted to list the 4 types of immunotherapies currently available. They are nonspecific immune stimulation, adoptive cell transfer. These also include TILs as well as CAR T-cell therapies, vaccination strategies, as well as the immunocheckpoint inhibitors. I wanted to emphasize again that I'm going to be talking the majority of today about the immune checkpoint blockade inhibitors. And I also wanted to add that these therapies are not mutually exclusive. They can be used in combination with other immunotherapies or conventional treatments to give even better results. Now, this is an idealized Kaplan-Meier curve adopted from Jim Allison's plenary talk a couple of years ago from the ASCO meeting. In immunology, or I should say in oncology, we're accustomed to measuring a control curve as outlined in purple. This is usually without drugs or treatment, and we measure that against a standard therapies outlined in blue, and we measure the difference. So that's the black arrow in between. And here we're plotting the percent survival versus time. So you want as many people to live as long as possible. So what we're looking for is a shift to the right. Now, the bigger the gap in between the two curves, the better. Along with the immunotherapies like ipilimumab shown in green, we actually saw a great response, but more importantly, we saw durable responses out several years. This was remarkable. This was the first time in cancer treatments that this has been noted. And when they gave combination therapies shown in red, say Nivo plus IPI, you even got better results. So remember that immunotherapy relies on the presence of antigens on the cell surface to stimulate T cells. This is the iconic graph from Mike Lawrence and Gady Goetz's Nature paper in 2013 that depicts the mutational burden of cancer in the 27 cancers of the TCGA, the Cancer Genome Atlas. Moving from left to right, you go from the childhood cancers with smaller somatic mutation rates, and they give way to adult cancers with the larger mutational burdens. These cancers, over on the right, like melanoma and lung cancer, are products of environmental exposure to carcinogens like nicotine and UV light. You can see down at the bottom the Circos plot that go from a circle without connections or rearrangements on the inside, to more and more chromosomal aberrations, indicating increased genomic complexity and pathology. When you have either a cancer cell or a vaccine, they're producing and displaying foreign antigens. These antigens can be on the cell surface or they can be in solution. Antigens are things that your immune system recognizes and mounts an immune response to. This can be either humoral or cellular. APCs, or antigen-presenting cells, will eat or phagocytize these molecules and display them as foreign antigens on the cell surface. And what they do is they actually educate the T cells to kill the tumor. This interaction is done via TCR or the T cell receptor. These foreign particles are what your T cell responds to. Now, the T cells are the most important cells in the immune response. When you have a cancer, you'll start activating your T cells that mobilize your immune system. These T cells will actually tell another set of T cells to go out and proliferate and start dividing. They'll recognize the tumor cells, and it should kill them. That is what normally happens, and it probably does happen for the most part. You get a small tumor or a small neoplastic event, and the immune system will clear it. You only notice when the system fails, and you get a pronounced tumor. The immune system then will upregulate actually a particular protein on those lymphocytes, those killer lymphocytes, called PD-1, PD standing for programmed cell death. Now, tumor cells are really clever, and they've figured out a way to get around this. So the cancer will get the kill signal of the T cell, and they get around it by making a ligand called PD-L1. So the PD-L1 ligand is displayed on the cell surface of the tumor cell. The PD-L1 binds to the PD-1 and turns off the kill signal of the lymphocytes. The PD-L1 is really a don't attack signal. This puts the immune system at a loss and opens things up for cancers to grow and to evolve. Now, the therapy that's come out recently that's very exciting is to block the inhibition of the T cells. So one of the most powerful ways is to do this is actually to use antibodies that recognize PD-1 on T cells. So the anti-PD-1 antibodies block PD-1 from binding to PD-L1. The tumor cell is then left vulnerable, and these effector T cells can now go and kill the tumor. These the PD-1 antibodies are very powerful, and in fact, there are several on the market, and there are more to come. This slide lists two of them, Nivo and Prim. Now, they've been used in a variety of different cancers, including melanoma, kidney, lung cancer, and I believe that the number is up to 15 different cancers that they've shown efficacy. This is a very clever way to open up cancer cells to get attacked by your immune system. Now, there's another approach that can be used, which is a general immune boost, and that is through the CTLA-4 inhibition system. We'll go back to our original graphic of the tumor shedding and presenting antigen. The T cell in this picture is in contact with the APC, the antigen presenting cell, and the T cell is generating stimulatory and inhibitory signals, as indicated by the green and the red arrows. Normally, the system is in balance between stimulation and inhibitory signals, and what we're trying to do is to push and pull the immune system in a certain direction. Now, the inhibitory pathway is CTLA-4 and involves a protein which is displayed on the surface of this particular cell. So this is an inhibitory signal that modulates the response of the immune system to keep it depressed a little bit, so it doesn't go completely out of control. You don't want your immune system to overreact to generate any self antibodies or a graft versus host reaction. Now, if we add antibody that is specific for CTLA-4 on the receptor, we can actually remove the inhibition of the T cell, and it then will go do what it was supposed to do all along, and that is recruit other cells that will eventually attack the tumor cell. So antibodies that recognize CTLA-4 inhibit this inhibitory system. The net result is a hyperimmune or a hyperactive T cell, which is something you really want with cancer. If you've got cancer, you're better off having a hyperimmune system than a depressed one. So these antibodies, again, inhibit the signal that is inhibitory. But they're doing it at a different point and on a different cell type than the PD-L1 antibodies. So the T cells get activated and enhances the whole immune system. This is effective in some cancers, but not all. And that's one of the mysteries about immunotherapies, is why it's working in some cancers, but not all cancers. So ipilimumab or Opdivo from Bristol Myers Squibb is one of those antibodies that's out there against CTLA-4. It's been used very successfully in melanoma. Now, I've told you about a couple of the biomarkers on tumor cells and immune cells, like PD-1, PD-L1, and CTLA-4. But what I didn't tell you was that there were a lot more biomarkers that people are investigating. These either upregulate the killer cell population or downregulate the inhibitory cells. So currently, there are 24 inhibitory molecules and 5 positive or stimulatory molecules that people are looking at as molecular targets. Obviously, if you start mixing drugs and targets, there are a lot of potential combinations to push and pull the immune system to react against cancer antigens. All of these potential targets are on different cell types, and I've graphed a few of these for you. So, for example, tissue macrophages that process antigen, the tumor cell itself, the regulatory and the effector cells, all of these have cellular receptors that can be regulated, either upregulated or downregulated, to give the desired immune response. And of course, there is a lot of business and a lot of financial interest in this area because cancer drugs can make big money. Here is a partial list of some of the immunotherapy companies. I'm sure that there are more coming. You'll notice that they're both established pharma companies like Bristol Myers Squibb, Roche, Merck, AstraZeneca that are on there, as well as a whole host of different startup companies. Now, there are a number of cancer immunotherapies in clinical trials. Here, I've actually broken it down into tissue type. The take-home message here is that there are many different cell types that are represented. So immunotherapy is against all cancers, or a lot of different types of cancers, not just one particular ones. Now, here is a short list, and I'm sure it's going to get a lot longer, of the the immune checkpoint inhibitors that are approved or currently in late-stage clinical trials, and probably will be approved within a year or two. You'll notice that all of these are monoclonal antibodies, as indicated by the suffix of MAB on the generic name. Now, one thing I'd like to point out is how immunotherapies are quickly being adopted into the standard of care guidelines. These are the new NCCN Guidelines released in March of this year for colorectal cancer. There is now a provision for giving either Nivo or Prim for dMMR, deficient mismatch repair genes, or for MSI high, that's microsatellite instability, high tumors. So the take-home message here is that immunotherapy is now being accepted as part of the standard of care. Now, in a related story, the FDA has granted accelerated approval to Prim for the first time as a tissue or a site-agnostic indication. This occurred a couple of months ago in May. The indications here are adult or pediatric patients with unresectable or metastatic cancer. that is either MSI high or deficient in mismatch repair genes. So all of those patients are eligible. Now, this particular indication is for solid tumors that have progressed. And this actually represents about 5% of colorectal patients will present probably with MSI high or deficient mismatch repair genes. Again, this is remarkable because it is tissue or site agnostic, and the FDA has never done this before. The second interesting thing about this study is that the number of patients that were used in the clinical trial was only 149. So this is remarkably few patients for the FDA to approve a major drug. So it really indicates that the FDA is embracing immunotherapies, and it probably reflects their thinking that the immunotherapies are lower risk than the current chemotherapies and targeted therapies. So remember that all of these tests that I've just mentioned reflect genomic instability and complexity. So if we go back to our graph that maps the frequency of somatic mutations per cancer, we can actually now overlay categories of tumor burden as measured in the number of mutations per megabase. As you can see, some of the tumors on the right-hand side are yielding 10 to 100 mutations per megabase. That's a lot. What this really means is that you're generating these neoantigens forming on the cell surface. So if you look at the graph or the figure over on the left-hand side, we actually have a tumor, and if we sequence it, we can identify these mutations. All of these mutations, or a certain portion of these mutations, will lead to changes in the protein, and some of these will actually present themselves on the cell surface. These new surface proteins are called neoantigens because the immune system has not seen them before. So the neoantigens are thought to be the target or the new target of the T cells. So any tumor expressing lots of neoantigens should, in theory, respond better to the immunotherapies. The more mutations in the tumor means more neoantigens, which means a better response to immunotherapies. So TBM, or tumor mutational burden, is emerging as a great biomarker for immunotherapy. Unfortunately, there are no specific rules or guidelines at this time for this testing, so people are still kind of developing the rules around it. Now, the laboratory tests for cancer biomarkers can now be separated into two classes. One is genotypic testing, and this is based on the specific nucleotide content. These can have specific genomic coordinates or can be variable. So examples of these are the classic BRAF V600E variant, or copy number variants, or any structural variants, indels, gene expression, which measures, of course, RNA, or any of the rearrangements or fusions. Also, mismatch repair genes will fall into this. The other new type of test is phenotypic in nature, in that it is not based on specific genetic variants, but is measuring a phenotype or a function or a characteristic of the cell. So examples of these include the MSI, the microsatellite instability, or the tumor mutational burden, the TMB. These are characteristics of the tumor and not of a specific gene. Here's a table of the lab tests that can be used to identify the possibility of neoantigens on the cell surface, and hence would make good candidates for immunotherapy if they're positive. MSI testing has been around for many years, and has been used for testing of Lynch syndrome and the susceptibility of inherited colon cancer. It is a PCR test. It's very, very simple. It takes about 5 or so mono or polymorphic repeat markers. You PCR those up and then run them out on a gel and analyze for stability of that region. It can also be you can also do that same test instead of a sizing gel. You could run it out on a sequencer and measure the fragment length. I just told you about the tumor burden TMB, so I'm going to skip over that. But I will say that a number of different labs are using it, are doing it today, and they're usually using large comprehensive NGS tests to make that available. Deficient mismatch repair genes, they're looking for either a germline or a somatic mutation in certain gene families associated with mismatch repair. Again, it is a characteristic of hereditary colorectal cancer. Remember that MSI and the dMMR mutations can either be germline or somatic. Now, one very exciting area in the personalized medicine space or the personalized cancer vaccine space is the vaccination strategy. Now, the goal here is to identify neoantigens or peptides that are unique to the tumor. So this starts with a patient with a tumor. The tumor is surgically removed or biopsied. The sample is then made into an NGS library and sequenced. Ideally, you would be able to sequence the entire genome, but there are several groups and companies that are doing this with just sequencing the genome or the exome or a subset of cancer genes. Now, the results from the sequencing are compiled and run through algorithms that will actually predict the abundance and the antigenicity of the predicted proteins, and if they're expressed on the cell surface. So you can actually go one of two ways. One is to determine if you want to give the patient standard generic immunotherapy, or if you actually want to go a more holistic way, and that is to personalize by synthesizing peptides that are predicted to be the most antigenic, immunogenic, and then either incubate these with immune cells through adoptive cell transfer from the patient or directly inject the peptides into the patient. So again, there are many options with regards to immunotherapy, and again, you can do these in combination, or you can do them in combination with conventional treatment. Hopefully the treatment will educate the patient's own immune system to respond to the tumor cell without attacking the normal cells. This is truly personalized medicine and a very clever way to fight cancer. So those are mostly the good things about immunotherapy. I did want to just have a slide or two on some of the bad side of immunotherapy. There are some side effects or common adverse events in treating patients with immunotherapy. This graph shows IPI, Primbro, and Nivo, and also IPI plus Nivo combination. And it maps these treatments to diarrhea, fatigue, nausea, pruritus, and skin rashes. Now, the drug makers are very much aware of these side effects, and they're paying close attention to any possibility of adverse reactions during therapy or during clinical trials, and they're monitoring these patients very closely. Another bad side of immunotherapy is the prices. Unfortunately, this is not positive. The cost of immunotherapies is very, very high. Here's an example of Nivo and IPI treatment alone being well over $100,000. and the combination of therapies being nearly 300,000. And I also should add that this price is per year. This actually brings up an interesting point, and that is we're still not quite sure of how to administer these drugs. So some people, some of the treatments have done 1 administration, 2 administrations, 3 administrations. It's kind of arbitrary. And there is research that's going on to sort this out. But it could turn out that you only need 1 dose of these very powerful drugs to essentially turn on your immune system, to educate it, and then you don't need any more. So that could be one of the good sides of immunotherapy. Again, one of the more important features of the immunotherapies and how they differ from existing therapies is that they can, and they are, being used in combination with the other conventional or immunotherapies. The conventional therapies, of course, being radiation therapy, chemotherapy, and the targeted therapies. So together with the other sorts of treatment, you can boost your immune system to attack cancer and at the same time be using the traditional targeted treatments, either drugs or radiation. So it's really a 1-2 punch. Obviously, the number of combinations in different immunotherapies and conventional therapies is very large. And of course, everybody will react differently. So it's going to be interesting to watch to see how the clinical testing of combinatorial therapies is done to get enough patients to actually prove that it is efficient. So I just wanted to end with one prominent success story, or at least so far it's a success story, and that is President Jimmy Carter. He was diagnosed with melanoma, and it spread to his liver and his brain. He underwent surgery, radiation therapy, and of course got this new kind of cancer treatment, immunotherapy. He underwent remission, and it seems to be, I shouldn't say cured, but they cannot detect any of the cancer in him today. Jimmy Carter was given Prim, which is an anti-PD-L1 antibody from Merck. It's going to be interesting to see how durable President Carter's remission really is. But for right now, he seems to be disease-free. So to summarize, this is a very exciting area, and there is a lot of hope as well as hype. Your immune system can be used to fight cancer, even the most aggressive kinds, and in fact, they work the best for immunotherapy. Cancer has figured out a way to block the immune response using PD-L1 ligands in many kinds of cancers. One strategy is to make therapeutics that block the PD-L1 inhibitory pathway. Antibodies from people are being used with the anti-PD-1 antibodies. They are binding to the T cell and releasing it from inhibition. Cancer vaccines can be used broadly on a number of people with common antigens, and for those people, if it works, that's great, but there's another backup therapy, and that is to actually personalize the treatment with peptides so that you boost the individual's immune response. Again, this is truly personalized diagnosis, analysis as treatment. This is really the way that cancer therapy should be. And what I'd like to do is show you my references that made this talk possible. There are some really great websites to learn more about immunotherapies. My favorite is shown on this slide. It is SITC, or the Society for Immunotherapy of Cancer, and you can go to this website and learn more. They actually have different areas depending on if you're a doctor, a researcher, or a patient. There's some great patient information there. There's also a new textbook which has just been published, and you can get, again, more information at the SITC site. So with that, I'd like to say thank you very much to the audience for tuning in, and I'd like to turn it back to Julie.
Thank you, Dr. Carmack, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, we will follow up with you via email after the presentation. Let's get started. Our first question is, how are CAR T cells involved in immunotherapy?
Yeah, so that's a very good question. So I didn't have a lot of time to go into the CAR T cell story. They're one of the promising areas of research coming along very strongly. So CAR actually stands for chimeric antigen receptor. These are genetically engineered cells to attack cancer. One CAR T cell therapy is expected to be approved this year for the treatment of children with acute lymphoblastic leukemia, or ALL. A second for adults with advanced lymphoma is probably also going to be approved either this year or early next year. It's still early for CAR T cells and other adaptive cell transfer methods. They're not quite as advanced as the immunoblockades methods are, but they're certainly showing a lot of promise. One question that people have been kind of asking is, do I use the checkpoint blockade inhibitors for solid cancers, solid tumors, and the adoptive cell transfer methods for hematological disorders, or can you mix and match them, essentially? Are both of the methodologies used against everything? And to tell you the truth, we don't know the answer to that. We're not quite sure what the sweet spot for each of the technologies is.
Thank you. The next question is, which companies do tumor mutational burden testing?
Yeah, so I believe Foundation Medicine was actually the first to report on the TMB, the tumor mutational burden, with Foundation One, and then they came out with Foundation Heme Test. So they actually report both TMB as well as the MSI, the microsatellite status. I know that there are other large comprehensive tests like MSK Impact that are also reporting this. There was a Nature Medicine paper from the group, the MSI, or the MSK group a couple of months ago that showed this. I think anybody can do this type of test with enough sequencing capacity. And I believe that there are some algorithms in the public domain to help calculate this.
Thank you. How is tumor mutational burden reported?
Yeah, so the TMB is reported as the number of mutations in a specific area of genetic content. So it could be reported in a couple of different ways, like mutations in a cell, mutations in a tumor. Most commonly, I think it's done as mutations per megabase, or area of the genome equal to the length of a megabase. So if they don't have a megabase of sequence, they may actually extrapolate it to give that number. Again, this is probably the most common way of doing it. How they make their cutoffs, I'm not quite sure. How that, if 10 mutations per megabase is enough, or 20 or 30 or where the cutoff is to be considered high or moderate or low. I'm not sure, and I think that's actually up to the individual company. So remember that all of the companies and labs are coming up with their own definitions of some of these terms. And depending on their design and their technology, there's not a consensus way of measuring it yet.
Thank you. We'll wrap up with one more question, and that question is, what is the difference between Nivo and Primbro?
Yeah, so they're not the same drug. They're both monoclonal antibodies that recognize PD-1, but they were generated independently of one another. Primbro is made by Merck, and Nivo is made by Bristol Myers Squibb. They're both whole antibodies, but Primbro is humanized from a mouse antibody and then grafted onto a human backbone, while Nivo was made from transgenic mice with transplanted human immunoglobulin genes. Then you inject the mouse and then you make a monoclonal antibody. They're actually different molecular weights, and I think that actually reflects that they're probably different classes of heavy chains. They're both kappas. I would guess that they're probably both IgG4s.
I would like to once again thank Dr. Carmack for his presentation. Do you have any final comments?
No, I'd just like to say thank you very much for attending, and this is a very exciting field. And to watch this area because there's going to be a lot more developments in immunotherapies.
Thank you once again, Dr. Carmack. I would also like to thank LabRoots and Vela Diagnostics for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 14th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.